with observations and so on, as you can imagine. And you also have to learn a lot about jargons in a given field. And what I will do instead is to do some basic tutorial of how to use equations for modeling biological processes. So for that, I chose this example of pattern formation because it aligns with the theme of this GEM4 school, which is about development. So I will spend maybe 15 minutes just to summarize these concepts of diffusion and reaction diffusion equation. Then we'll switch to the blackboard to introduce the key concepts of this Turing mechanism. Anyone heard about these Turing patterns and so on? No one ever heard about this yet? Yeah, we'll see. And if, depending on how well you follow this, you may or may not have time to talk about these molecular motors that are responsible for carrying things or generating forces in these organisms. So biology is full of wonders, and one of the wonderful things about biology is this regular pattern formation. Means, say you are taking some cells from the skin of this fish or some proteins. They have no idea what pattern they are going to make. They are just interacting locally with their neighbors. Then how can this long-range order be possible through these local interactions? That's something we are going to talk about. And by the way, for my lecture, we'll use some undergraduate level mathematics. And are there any biology major students who have never taken linear algebra or something like that? Everyone knows matrix, right? How about eigenvalues? Yes? Yeah, that's pretty much all you need to know. Okay, so let's take one protein from this fish, and we'll model it in a very simplified manner. And it will undergo diffusion or Brownian motion, and we'll consider its motion in 1D. So it walks left and right with step size L and equal probability. So there's no bias in walking. And this is a stochastic process. So every time you do it, the particle will move in different ways. So let's ask this question, where will that be after making n steps? And again, this is a random event. So we introduce this so-called random variable, Si, which denotes the ith step. So this variable Si has value either plus L or minus L with probability 1/2. So we can express the position after making n steps as just sum of these Si's up to Sn. Is that clear so far? Yeah, so then we can, for example, calculate the average position. We denote the average in this angular bracket. So following this expression, the average position is simply the sum of the averages. And average of Si can be easily calculated. You have this value times the corresponding probability, and you sum over these probabilities, then it becomes 0, of course. There's no bias. So on average, the particle doesn't go anywhere. But if we know that if you put many of those particles, they will spread out. So how do we calculate the spread? We use this variance, which is the mean square deviation from the average. You are all familiar with variance, right? Yeah, so in this case, the average position is 0. So the variance becomes simply the average of the square. And again, we use this random variable Si's and replace X with the sum of Si. Then we expand this product to get S1 squared, S2 squared, down to Sn squared. And we also have all these cross terms, S1, S2, S2, S3, and so on, which is this. So if you make an average, Si squared average is again, easy. You have the value squared plus L squared minus L squared multiplied by the probability. You get just L squared. So the first term gives L squared summed n times, which is nL squared. And the second term becomes 0 because each of these steps are independent. So the average of the product becomes the product of the average, which is 0. So we arrive at this important basic relation of diffusion where variance grows proportional to the number of steps. So when you express the extent of spread, you take the standard deviation, which is the square root of the variance. That grows as number of steps to the 1/2. So suppose you make 10 steps and you spread. Then you make 100 steps. Your spread doesn't become 100-fold. You spread only by 10-fold. And when you make 10,000 steps, you don't spread that much. You only spread 100-fold. So the diffusion has a property that the efficiency of spread becomes less and less as you make more and more steps. We will come to this point again. But right now, let's relate this number of steps to time. Sorry. I think yeah. So that can be done easily by just measuring how long it takes to make a step, the typical stepping time. So you can have this relation that t is equal to number of steps times the stepping time. And I mean, if you work out the detail and the coefficients, you get this x squared average going as 2dt. And we'll use this extensively in appreciating some of the results later. So d is the diffusion coefficient. And from this relation, we know this dimension is meter squared divided by time. You just divide by time, then you get the diffusion coefficient. And diffusion coefficient also denotes this ratio between thermal energy and drag. So if there's more thermal kicks, the particle will diffuse more, which is proportional to the temperature. And if the viscosity of the medium is higher, then the thing is expected to diffuse less. So you have this ratio going up as the diffusion coefficient. And again, if you work out the detail, you get the coefficients like this for a spherical particle. And this is the famous Stokes-Einstein relation, which Jeff Fredberg mentioned yesterday. And physically, this step size and stepping time are related to the mean free path between these random collisions. And the time between the random collisions sometimes is called the correlation time in this Brownian motion. And note that when you go inside of a cell, the drag part becomes higher because of this macromolecular crowding. So when you measure the diffusion coefficient of your protein in solution, that's usually higher than the diffusion coefficient in the cellular environment. So because of that, it's harder to diffuse in the cell. And let's first of all compare diffusion with the advection. I mean, advection is a transport with constant velocity, usually by motor proteins. So the like x grows as v times t. So if you plot this delivery distance versus time, advection grows linearly in t, and diffusion grows square root of t because of this L squared going as d times t. So as you can see here, in short distances, diffusion is more efficient. So diffusion is sort of faster in short distances, and it becomes slower and slower in long distances. So when does this crossover happen? So we just set some distance L, then time to diffuse this distance we can express as L squared divided by d. I mean, we are going to ignore all these coefficients, like 2 or something. So this is a diffusion time. And we can analogously, likewise express the advection time. which is simply L divided by V. So this ratio between these two times, TD and TM, that becomes VL over D. It's called the Peclet number. Have you heard of this number, Peclet number? Okay, so if this number is greater than 1, that means TD is greater than TM, so diffusion is less efficient. I mean, you can see the dependence. If the molar velocity is higher, then it's easier to have this diffusion less efficient. And if the direct coefficient is, diffusion coefficient is less, then this number grows. Then again, diffusion is less efficient. So if we take like an example of like a globular protein of size of like 2 nanometers, then the crossover happens around the size of a cell. So for these small proteins, they don't really need motors to be carried over within this cell. But when you consider vesicles or larger cargoes, then this crossover distance becomes shorter, so they definitely need more proteins to be carried out. I mean, especially when you talk about like axons, neurons, and so on, they are centimeters in length. In that case, diffusion becomes hopeless. So you really need a motor. And this electron micrograph shows an axon, and this is the microtubule, and this is a vesicle, and you can see that there are motor proteins attached to this vesicle. I mean, schematically drawn here. And by the way, any idea what these are? Microtubule is here, which is thicker than these filaments. They are actin filaments. What about the things that reach these actin filaments? Yeah, they are likely alpha actins. Okay, so motor proteins can carry cargos, but they can also generate diffusion-like behavior. And since there are many motors attached, some of them may walk to the right, some of them may walk to the left. So then this can become a tug of war. So there will be some times where these motors work, and another time these motors walk. So we can call this switching time between these two motors as tau m. Then this cargo basically does this diffusive motion, I mean, in an effective sense. And we can estimate the diffusion coefficient in the following way. Again, we'll use this L squared going as d times time. And now L is the distance of travel while one type of motor is engaged. So that becomes V times tau m. Vm is a motor velocity. So you can estimate that the diffusion coefficient in this case scales as motor speed times the switching time. Maybe some of you remember this violation of fluctuation dissipation theorem during yesterday's lecture, right? So in this case, diffusion coefficient doesn't follow Stokes-Einstein relation. It has a different scaling form. But details of this non-thermal behavior is not very well understood. But what this example shows is, I mean, although diffusion originally was conceived as from this thermal motion, it can be used for non-thermal motion, random motions of motion of growth factors, proteins, and cells, and so on and so forth. Okay. So far, we just considered a single particle motion, and now let's move to multi-particle motion. Then, of course, we use this density to describe the motion. And from our everyday experience, we know that particles move from denser to sparser region. But actually, what I just said is a misnomer. Without bias, there is no motion. I mean, we have to really talk about this flux. So flux is the number of particles passing through a given point per unit time. And from this intuitive behavior, we can see that the flux should be proportional to minus of the concentration gradient. higher to lower density, negative. And we can write it in this compact form. Here, partial x means partial derivative relative to x. And this proportionality constant turns out to be the diffusion coefficient we introduced earlier. And this relation is called the Fick's law. Have you heard of Fick's law, right? Or no? Yes. Okay. So likewise, we can consider how the concentration depends on time. And if we consider this thin slice of thickness dx, there is an input of particles from the left with flux Jx, and like a particle leaving this region with flux Jx plus dx. So if these two fluxes are different, the concentration in this region can change. So mathematically, you write this as the partial derivative of c relative to t equal to the negative gradient of flux. And if you use this Fick's law, you arrive at this famous diffusion equation. Partial t of c is d times partial x squared c. And if this particle undergoes some chemical reaction, you just add reaction terms to the right-hand side of this equation, and that becomes a reaction diffusion equation. And more formally, it's called the Fokker-Planck equation. And there is even a book with the name Fokker-Planck equation. It's a very well-developed area. So before we switch the blackboard, let's just work on one example of this one component system, generated at the source, and it diffuses out, and it's degraded with rate k. Then we can write this process in the following way. We have this diffusion equation, and you add the degradation term, minus kc. And after a long time, steady state profile is formed, and what we are interested is how wide this region is. Then steady state means the thing doesn't depend on time anymore, so you set this time derivative equal to 0. Then the remaining equation just becomes a regular second order differential equation. So a function whose second derivative becomes itself with certain coefficient, that's an exponential function. So we have this profile, and we can even get this this decay constant as the square root of the diffusion coefficient over this degradation rate k. And we can understand this relation even without solving this reaction diffusion equation by considering 1 over k as the average lifetime, right? Because the degradation rate is k, 1 over k should be the lifetime of this particle. So this relation is nothing but lambda squared going as d times tau. Again, application of the same simple fact. So even if you are not going to work on diffusion equation, this is something you want to remember. Length squared going as d times time. And this is an example of a Morphogen gradient called decapentaplegic. I'm not very familiar with this term. It's a Morphogen, I think it's related to like BMP, bone morphogenesis factor or something in Drosophila. So shown here is really the exponential profile, as we have seen here, for different embryos. They all follow the same line. So in this way, you can estimate what the diffusion coefficient is, or what the degradation rate is, and so on about these particles. What about multi-component system? Right now we just talked about a single diffusing species. So multi-component system, our tutorial begins. So the main idea of this Turing patterning is instability driven by diffusion. So otherwise the system will stay stable, but the novel idea by this Turing was that diffusion can give rise to instability, which is fairly counterintuitive. And diffusion usually smooths these things out, but depending on the case, sometimes instability can be developed. So what's the condition for this instability? In one sentence, it's short-range activation, long-range inhibition. And we'll see what we mean by this now. Any questions so far? Okay. So let's go offline and we'll move to the blackboard. Is there any way? Okay. And how do we move that up? Or blank? Oh, great. Then okay. So I said we'll work with the two-species system. Can you all read this from the back? Okay. Then we write the reaction diffusion equation. Diffusion term plus some reaction term. Now that involves two species. And we can also write for the second species. These two depend on the system or model. So right now we'll work on the simplest possible case where things are linear. So F C1 C2 is let's write alpha 1 C1 minus beta 1 C2 plus gamma 1. And G C1 C2 is alpha 2 C1 minus beta 2 C2 minus gamma 2. So what do I mean by this equation? We can write it in more pictorial way. Here's species 1 and 2. 1 is created through this term and that term. So alpha 1 C1 is kind of a self-generating term. And there is an injection of particles with rate gamma 1. And 1 is eaten up by 2 through this term, which we use by this symbol. So it's called inhibition. And if we go to species 2, it eats up 1 and it grows with rate alpha 1, alpha 2. And it decays with constant beta 2. And it's removed with rate gamma 2. And both of them diffuse with diffusion coefficients D1 and D2. And we can call from this relation 1 as the activator and 2 as the inhibitor. Fine so far? Okay, so we need to solve this. I mean, most of people, when they see equation like this, these days, they just feed this into MATLAB or something like that and just get the solution. I mean, that's good, but that doesn't tell you the insight about the system. So to develop insight, we need to use our hands. I mean, that doesn't change over time. So now let's start to analyze this system. And because this looks fairly complex, we will be a little more simple. So we first consider without diffusion and steady state. So without diffusion means D1 and D2 are set equal to 0. And steady state means the time derivatives are 0. Which means we just set f and g equal to 0. So when you set f equal to 0, we get C2 equals to 1 over beta 1, alpha 1 C1 plus gamma 1, and G equal to 0 gives C2 equal to 1 over beta 2, alpha 2 C1 minus gamma 2. Right? So C1 and C2 have to satisfy these two equations in order to reach the steady state. Okay? So we'll solve this in more pictorial way. It's like y equals something x plus something. Straight line. Again, the same in C1 and C2 space. So we plot this axis of C1 and C2. By the way, all these coefficients are set to be positive. So the first graph, let's call this as A, and the second graph as B. The A graph has intercept with the C2 axis on the positive value. So we have this for A. And now we plot this second graph. It has intercepts in the negative value of C2 axis. And if this line is parallel to the line A or has slope less than A, these two graphs do not meet, which means there is no combination of C1 and C2 that can satisfy this. So anything that's parallel to this or lower, there's no solution. Is this clear? So in order for there to be a steady state, the slope here must be greater than the slope there. So it has to be like this. So let's call this point as C10, C20. So we started out with this linear equation, but now we know one condition for steady state. For steady state, we should have the slope here greater than slope there. So alpha 2 over beta 2 greater than alpha 1 over beta 1. So from this we have alpha 2 beta 1 minus alpha 1 beta 2 greater than 0. So that has to be satisfied to get that. But finding steady state is not the end of the story. I mean, when these particles, they can be morphogens or genes or whatever, when they start out at some point in this phase space, their initial concentrations are here. They have to be able to reach this steady state to establish a profile. So we need to find the condition for that. I mean, they might just move away from this steady state, then unless you start right at this point, you will never reach this point. So we'll analyze this sign of time derivatives here. Now D1 and D2 are 0 right now. So we'll draw this and see when, how this C1 and C2 behave. So along A, del T of C1 is 0. Along B, del T of C2 is 0. And suppose you are above this A. Then C2 is greater than what's necessary. So here, if C2 is too big, then F becomes negative, which means del T of C1 becomes negative. So above this line, del T of C1 is negative. That means you have decrease in C1. Below line A is the opposite. Del T of C1 is positive. Okay? How about across this line B? If you are above this B, then C2 is greater than what's necessary. So G is negative. So C2 decreases. If you are below line B, the opposite. So we have this behavior around this steady state. Is that clear? Yeah, please stop me at any time if you feel something is not right or if you don't understand. So let's take an example. We have this, we have that. Suppose you started from this point, some arbitrary point. Here, C1 decreases, but you are below line B, so C2 increases. So you go like this over time. And if you reach this point, C2 doesn't change, but C1 still decreases, so you move out. Then once you cross the line B, then C2 starts to decrease, so it has to actually be like this. So you reach this line, then if you cross this line, C2 still decreases, but now C1 is increasing. So you go like this. And similarly, once you cross this line, C2 and C1 increases like this. So the behavior becomes like this. And depending on the magnitude of these coefficients, you could just circulate like this forever. In this case, if you move away from steady state, you will never reach the steady state again. You'll just keep on oscillating. So this is the case of oscillation. Or if these coefficients happen to be such that you just keep on moving away, so you spiral out, that's unstable case. So only when you fall like this, then this steady state is a stable steady state. When you start out from a combination of concentrations, you reach this point at long time. So this spiraling in is called the stable case. So you have these three different regimes of behaviors depending on these parameters. We talked about these waves and oscillations and so on in gene expression a little bit during Professor Keller's lecture. This partly captures those processes. I wouldn't say this is all that's needed, but it shows when things can be possible and when different behaviors can arise. So we had this one condition for steady state. Now we need to find the condition for stability. So what we can do to study this stability is we move away a little bit from this steady state and see if we just keep moving away or if we return back to the steady state. Which means we want to see the behavior of the deviation from the steady state. If this delta C1 and delta C2 keeps on growing, then the solution is unstable. Okay? So let's take the time derivative. That is just del T of C1 minus del T of C1 0. And what is del T of C1 0? Yeah, it's 0. It's just a constant. So it's just del T of C1. And similarly, del T of delta C2 is del T of C2 itself. And we'll be a little bit more formal. Del T of C1 is now F of C1 and C2. We are ignoring diffusion so far. Then we can write things in terms of delta C1. C2 0. We just replaced C1 and C2 using delta C1 plus C1 0 and so on. So whenever you see something like this, some major number plus a perturbation, what we can do is, it starts with T. Yeah, Taylor expansion. So we can write it as F of C1 0, C2 0, plus delta C1 or del 1 F plus delta C2 del 2 F. And F of C1 0 and C2 0 is 0. That's what we initially impose for the value of C1 and C2 0. So this becomes simply alpha 1 delta C1 minus beta 1 delta C2. And the constant terms do not count after partial derivative. So we started with this rather arbitrary expression, but actually these numbers do not matter. All that matters is that they cross. So whether or not you vertically shift these graphs, that doesn't matter. So likewise, you can write del T C2 as alpha 2 delta C1 minus beta 2 delta C2. Okay? So this looks a little messy. So we need to have ways of simplifying things. Let's write it more in a tidier manner. Sorry. Alpha 1 delta C1 minus beta 1 delta C2. So now the things look a little better, and we can introduce a simplified notation. W as a column vector of delta C1 and delta C2. then we can write the whole equation like this. Time derivative of this vector W is a matrix times W. Have you ever seen things like this before? Okay, great. So what is A? It's a 2 by 2 matrix. It's just these coefficients. Alpha 1 minus beta 1, alpha 2 minus beta 2. Okay? Okay or not okay yet? Okay. So you have something that looks like y dot is ay. This has the same form as that. Then what's the solution? You have first derivative, not the second derivative. Y goes as e to the at, simply. So following this analogy, I'm not going to work on details of this matrix, but we can say W grows as e to the some number times t. And any guess of what this number should be? Lambda actually becomes the eigenvalue of A. I mean, that's what I asked this at the beginning. So we just need to solve the eigenvalue equation, then we can see the time dependence. So let's solve the eigenvalue equation. A minus lambda I is the diagonal matrix, identity matrix. That has to be 0 in determinant. So we have alpha 1 minus lambda minus beta 1, alpha 2 minus beta 2 minus lambda equals to 0. So let's write that. We have multiplied this, subtract that. So we have lambda minus alpha 1, lambda plus beta 2, plus alpha 2 beta 1 equal to 0. So you expand this, lambda squared plus beta 2 minus alpha 1 lambda plus alpha 2 beta 1 minus alpha 1 beta 2 equal to 0. And this is in the form lambda squared is equal to p lambda plus q. So the solution is 1/2 minus p plus minus square root of p squared minus 4q. Maybe we won't do all this algebra. Let's just take a look at the signs. Q is this one. Is it positive or negative? We got this for the condition for steady state. So Q has to be positive in order for there to be a steady state. So you have p squared minus a positive number. So the whole thing is less than p in terms of magnitude. So lambda becomes negative p plus minus a number that's less than this one. So in order for this equation to be stable, all eigenvalues of lambda has to be less than 0. Why does it have to be less than 0? Because of this. If lambda is positive, your perturbation will grow in time, and your perturbation is the distance from the steady state. That means a positive eigenvalue of the matrix will lead to your solution moving away from the steady state. So to satisfy this, we must have p greater than 0. Which means beta 2 greater than alpha 1. So we are deriving very general conditions for steady state and stability. And although we started with linear cases, when we expand, Taylor expand f and g, actually we don't need to have these to be linear functions. All that we need is the first derivative and their coefficients. That's why this linear approximation is good for studying this instability, at least. So we can be more complex, actually. But these lessons or conditions apply in general. So having worked out this, let's move on to the case with diffusion. And now we don't need to work out things like this. We can just directly write something like this. So we write del T of W equal to A times W plus D matrix W with the matrix D given by D1, 0, 0, D2. Does that make sense? And there has to be double prime. Okay? Yeah, then we are almost the same as this situation, except that we have double derivative here. So cases like this can be treated using the Fourier transform. So WK is integration of dx e to the ikx times wx. And actually there is time dependence too. So if you apply Fourier transform here, this equation becomes del T of WK equals A W. And again, I'll skip a little bit of detail in integrating this double derivative. Basically, the double derivative brings this ik twice. So we have ik squared, which becomes actually minus the k squared w. Okay. So now we have in the Fourier space the same expression like before, except we have different matrix in front. So let's find the solution of this form. Again, lambda is the eigenvalue of this matrix now. So this matrix is like this minus d1, d2 k squared. Like this. We are following the same analogy as here. Okay? Then, I mean, we can expand it in the same way you just multiply these terms, or subtract multiplication of these other terms. So we have lambda minus alpha 1 plus d1 k squared times lambda plus beta 2 plus d2 k squared. And you have plus alpha 2 beta 1 equal to 0. Then let's just expand this. Lambda squared plus d1 plus d2 k squared plus beta 2 minus alpha 1 lambda. And you multiply these two. d1, d2, k fourth, and d1 beta 2 minus d2 alpha 1 k squared plus alpha 2 beta 1 minus alpha 1 beta 2 equal to 0. So we are following almost the same procedure. As here. So we write that in the form of lambda squared is p prime lambda plus q prime equals 0. And again, the solution is 1/2 minus p prime plus minus square root of p prime squared minus 4q prime. And without d1 and d2, we said we are in the stable case. So the solution is stable. And now we are going to find the condition where introduction of d1 and d2 leads to instability. If this eigenvalue is greater than 0, then we have exponential growth of solution away from the steady state. So this is a condition where instability can happen. So again, let's just take the sign of these things. So Q is given here, alpha 2 beta 1 minus alpha 1 beta 2. And yeah, we have all these stuffs. So let's see. K squared. Yeah, first of all, P is given here. This is P, P prime, and that's Q prime. And here, the first term is positive. The second term is also positive because of this condition. So P prime is always positive. Let's go to Q prime. This term is positive. Those terms, that term is also positive because of this steady state condition. And the only thing where the Q can change time, the sign is here. But if we have P prime less than 0, the only way where lambda can be positive is this number to be greater than that. So does that mean Q positive or negative? Q prime. It has to be negative to be greater than this one. So in Q prime, the only place where sign can change is this. So we must have d1 beta 2 less than d2 alpha 1. But we learned that beta 2 has to be greater than alpha 1. So the only way this can be satisfied is by having the inhibitor diffusing faster than the activator. That's what we mean by short-range activation, long-range inhibition. Inhibitors can diffuse faster then they can inhibit growth of the activator. So what we did right here is just introduce basic conditions where diffusion can introduce instability. And the key is this difference in the diffusion coefficient. So any question so far? Okay, so then let's go back to the screen. Okay. So when we went, initially had this review paper, this fish and so on. That paper has a supplementary information, which is a Java simulation. So, we have an equation very similar to what I described. We have linear equations and the diffusion terms. And if we start with completely homogeneous case, then diffusion won't work. So in that case, if you run the simulator, the simulator just integrates this equation. You have this oscillatory behavior leading to a steady state, which is what we drew on this C1, C2 phase space. So you have this stable and steady state. But if you have random distribution of this activator and inhibitor, then over time, instability develops and you start to form these patterns. Note that they have no idea of what pattern they form. They just interact locally, but still patterns can emerge by some subtle choice of parameters. So let's say we have an initial condition like this. You run it. Then this wave propagates and the profiles start to form. We are using exactly the same equation. We just have different initial condition. And we said D2 has to be greater than D1. D2 is here, 0.5. D1 is 0.02. And let's change that and see what happens. Clear? Let's say we make this as points like 8. Maybe it's too small. Maybe it's too big. Let's see what happens. So now we have this initial condition. Run it. Oh, the inhibitors are diffusing too fast, so nothing much is propagating. So we have to reduce the number. That was 0.5. Let's say 0.8. Yeah. Then you develop rings like this. And if you notice, the size of this ring became wider than the previous case. And so on and so forth. And another condition we imposed was beta 2 greater than beta 1, which is down there. So here what corresponds to beta 2 is like this beta 1, I think, is like that. So we can control this and see what happens. Beta 2, maybe 0.15. Let's see what happens. Beta 2 describes the degradation rate of the inhibitor. So inhibitor just died away in this case. So even using the same set of equations, you can obtain a lot of different cases. So although this doesn't tell the whole story of this pattern formation, it kind of gives an insight of what can happen.
What if D1 equals D2? At the point where the diffusion is the same.
If they are the same, then there will be no instability developed. Let's see what happens, actually. So D1 was 0.02. D2, let's make this one 0.5. And you do this. It became just homogeneous. Yeah, there's no instability. Any question? Okay, so then we can have now two choices of what we'll do now. We can just stop here and do like question and answers, or we can go like maybe 10 minutes more to talk about these motors. What do you want? Motor? Motor? Okay. So maybe that's a good choice. So this is a figure I drew actually for a review paper I wrote with Matt Lang. And I drew things to the same scale. So these are actually relatively the right size in the cell. What this shows is a collection of motors, nucleic acid bound motors, polypeptide bound motors, and cytoskeletal motors. And these are the corresponding tracks. They vary greatly in terms of their sequences or families and so on, but they must have evolved under one physical constraint. They should be able to attach to one of these tracks, and they need to generate force and they move. So what design principle must be there to generate this directional motion? I mean, it's an enormously complicated system, and we'll just work on this smallest known processive motor, kinesin. Kinesin works on the microtubule. So this movie from the Vail lab shows the cargo transport, which we talked about. The vertical size is 13 microns. It's a real time. And you see these vesicles and so on just being carried by these motors. Most of them know where they are going. But sometimes, let's just wait for a bright spot. I think one will emerge from here. Sometimes they get confused. Maybe this one. Let's see. Yeah, maybe this one. Oh, this just, maybe this one there. It goes there. Oops. Yeah. Yeah, I mean, if you pay attention, you will see things going back and forth, which is like this active bias diffusion. So what's happening inside? So another case is this cell division. I mean, you may have seen this mitotic spindle picture many, many times. But if you pay attention, it's really dynamic process. Here's the kinetochore microtubule. They don't just get stably attached to this microtubule organizing center. They constantly detach and attach, which is organized again by these kinesin motors and some other motors as well. So below this movement, there are all these motors at work. And here I'll just compare these two types of kinesins. Kinesin 1 is this microtubule plus end directed motor. it walks processively, which means it walks hundreds of steps on microtubule before detachment. So it's good for transporting cargoes. Works as a solo motor. Kinesin 14 that includes this NCD motor is oppositely directed, microtubule minus N directed, and after making one step, it detaches. So it has to work as a group, which is well suited for organizing mitotic spindles like this. And we'll see what other basic principles of their operation. And this is an animation of the way how kinesin is believed to walk. So one motor head binds to the microtubule. Then that leads to ATP binding. And ATP binding triggers this walking step. And ATP hydrolyzes from this head, which detaches the guy. That allows the binding of the next ATP. So this has to be tightly controlled. And the next step happens, and so on. And remember, this is just an animation. It doesn't show what physical principles drive that motion. And more like confusing thing is when you compare the crystal structures of kinesin 1 and NCD. When you overlap them, they have almost identical motor head. The main difference is in this neck region. Neck is the region that connects the motor head to this neck stalk, and there's a cargo at the end. So kinesin 1 has this red flexible neck linker. NCD doesn't have neck linker. It just has this neck coiled coil continuing down to the motor head. So we just call it the neck. It has a neck alpha helix. So neck linker, sorry. So if you swap these domains between these two kinesins, you can actually make these walk in opposite directions. Although the motor becomes impaired, still it's possible to see the opposite motility. So it's been known that these domains are important for direction determination. But again, how they do that? So in the case of kinesin 1, the guy who walks to the plus end, I did simulation and found that the force generation is by this dynamic disorder to order transition. In this case, where one motor head is bound and this guy is loosely attached, then this allows ATP binding. Then the neck linker and the N-terminal flexible strand called cover strand actually forms a beta sheet. And the beta sheet has enough conformational bias to move this guy forward. So now you'll see result of a simulation, not just animation, showing this semi-deterministic stepping-like motion. And the red here is the cover strand. Before my work, no one paid attention to this domain. But if you delete this domain, then you can, basically this motion doesn't happen. And I also calculated how much force can be generated by this beta sheet, which is enough to account for the experimentally measured force. So that's about simulation. How do we test that experimentally? Oh, sorry. Since this cover strand has been unknown to the kinesin community, we designed mutants. Like the 2G mutants introduce 2 glycines to make this cover neck bundle more flexible. So this one supposedly has less force than the wild type. And in the Del mutant, we deleted out this cover strand. And the single molecular experiment was performed in Matt Lang's lab. So this is one example showing the stall force histogram. The wild type stalls at around 5 piconewtons. The 2G mutant stalls at around 3 piconewtons, which is in agreement with our predictions here. And the Del mutant stalls near the force detection threshold of their optical trap apparatus, which means within the experimental error, this guy really doesn't generate force. So this way we can control motility. How about NCD? It's a little more complex. I mean, there are 2 types of crystal structures known. One is in the pre-stroke state. One is in the post-stroke state of the neck. So the neck does this lever arm-like motion, but nothing much is known in between. That's where simulations can fill in. So I'm just going to show you one simulation trajectory. of the neck moving from pre to post stroke position. And note that this is not the way how entities actually move. It cannot be this smooth. The actual motion will be very like random and thermally agitated. And what we show from the simulation is the possible minimum free energy path, the most likely path where entities will make a stepping. So what else can you get from simulations like that? First of all, we find the neck motion is not just smooth swing. It makes this series of intermediate contacts. This is a list of contacts. This is the distance of travel from the pre-stroke position. So there is an intermediate context. So the neck motion is not just diffusion, it's a guided diffusion. As the neck moves, the motor head holds the neck all through this travel. And why would that be necessary? Remember, these guys have to walk against load. If this motion is just pure swing, if there is a load, this guy won't move. But if there's an intermediate contact, then by thermal fluctuation, you have a little bit of motion is held, and so on. So then you can work against the load. So that's like an efficient design strategy. And another interesting point was when we measured this free energy change for the forward and reverse motion. When you go this way or that way, the free energy change actually is different, meaning that this guy is showing energetic hysteresis. So why is hysteresis needed? I mean, note that this guy is working in out of equilibrium. So if this neck moves just in the equal way, there won't be any directionality. But by using this hysteresis and by using ATP, you can pretty much rectify its motion in one direction. So if you combine this guided diffusion and hysteresis, you can generate a directional motion. So although we just studied two families of kinesin, we learned some fundamental pictures, like disorder to order transition. I mean, before people just try to look at the crystal structure, which is a static snapshot, and try to figure out how force is generated, which is not possible because it lies in the motion itself. So this dynamic disorder to order transition, or this guided diffusion and hysteresis. I mean, these are the basic concepts we developed, and I think they are applicable when we analyze other types of motor proteins. They have to have such components in their dynamics, which we can look for now. Any question? Okay, so I will just close by citing some of the statements that Alan Turing made in his seminal paper. By the way, this year is the 100th anniversary of Alan Turing's birth. So I think it's a nice time to honor his pioneering contribution. I mean, these days, people use a reaction diffusion equation really too much beyond what they can be applied, which Professor Keller mentioned. And modelers just use whatever they think is right, but you have to be very careful, which Alan Turing was. So he knew that this is not a simple problem. You have physical laws, you have combination of biology, mathematics, and chemistry, and so on. So we are dealing with truly multidisciplinary problem. You can't just answer one question by working on one area. And down the line, he also knew the importance of mechanics. But at that time, they didn't have enough experiments and enough computational power, so he just set this aside, and he just focused on the chemical basis. But sometimes when a theory becomes effective, people forget about what the initiator thought, and they just apply the results blindly. So you have to be careful about using equations. And this is another amazing part. Remember, note that this is 60 years ago. He was talking about the potential help of digital computer. And nowadays, you really have the power, and we can address a lot of questions at the molecular level. These themes. So finally, there are some references. I mean, I guess you'll get this slide. And these two are nice review papers about this mechanics in morphogenesis, or this reaction diffusion approach. This is a thick book. You may not have enough time to read this book, but this book is really resourceful when you deal with a reaction diffusion equation. The next two books are very challenging books, but they are like gold standards in understanding stochastic processes, which biology is. So, I mean, I suggest, if you have the motivation, I suggest you to read this in your spare time. It will take maybe years to understand these books, but it's worthwhile effort. Okay, I'll stop now, and if you have any questions, I'll be happy to answer.
In your case, you're using linear system, but in most biological systems, they are non-linear.
Exactly.
So how should we address that?
Yeah, so the linear approximation works only for testing the case of instability. When you are away from your steady state a little bit, what happens? But what happens we cannot answer. We can only answer in the linear approximation whether it becomes unstable or stable. So if you want to know the full development of the pattern, you have to fully work with the nonlinear case. Then you can use computers to carry out the calculation. But analysis like this will give you insight or conditions, the bare minimum conditions. So you have to be careful. I mean, this is not the end of the story. This is just the beginning of the story. Yeah.
Researchers studying mechanism, usually they change the region's length.
Yeah.
But you apparently studied the pocket region that's important in the that motor protein as well. So did you also consider changing the length of your region of interest or modify?
Actually, we didn't change the length of the neck linker. I mean, that has been done by many groups like Bill Hancock or Steve Block recently did that. And it's not just the tension on the neck linker that controls this kinesin behavior. Especially Steve Block's recent paper in Nature says the orientation of neck linker is also important, which is not well understood at this point. And what we instead did was to mix and match this neck linker and cover strand of kinesin 1 and another kinesin 5 from mitosis. And we are finding some interesting compatibility and versus incompatibility between these regions. So we did some different mutagenic analysis. Yeah, question?
Can you say a few more words how realistic are these models in terms of when we apply it to explain quantum in statistical
Okay.
snails or the patterns in birds and feathers?
Yeah, that's an excellent question. I mean, not everything can be explained, but just this picture shows how accurate you can be. This is the real thing. This is the result of simulation. Real thing, simulation. Real thing, simulation, and so on. So it can be pretty accurate, but I mean, we didn't consider the role of force here. And suppose you have some embryo develop or something like that. There's a morphogen that commands cell division. So the size of this cluster grows. When do they stop? That's a big question. And there's a recent paper by Boris Schreiman at UC Santa Barbara saying that this control of this embryo size is actually mechanically governed. Because, I mean, if you follow just the Morphogen Gradient and so on, it just doesn't match with what happens. But if you think when cells divide, then there will be internal stress developed because there are just more cells crowding. Then cells will feel the force. Then if they feel some force, then they can generate force-dependent signal locally. So there doesn't need to be any global hand controlling the cell, the size of the cell. it can be just like locally controlled, as we just talked about right now.
Can you give us the reference?
Oh, yeah. Actually, I think it should be on my computer. Just a second. Well, I don't know. So, yeah, it's by Boris Schreiman. I'm not sure if there is another N or not. It's PNAS in 2000 something, maybe 2010 something. Yeah, it's at UC Santa Barbara. And mechanical force is actually a very good signal for controlling this whole thing. Because when you secrete signaling molecules, they have to, diffuse or they have to get carried out. But mechanical signal, they propagate at the speed of the sound in the tissue. So you can get very quick distribution of signals by force. Yeah.
So when you show the examples of the simulation,
Yeah.
I see depending on the conditions of the parameters, you can generate the patterns.
Yeah.
But it all depends on the initial condition, actually.
Also depends on the initial condition, yes.
I'm just wondering, when they actually simulated the patterns from the real cases,
Yeah.
I mean, it really depends on how they start, like where they start from the initial conditions. I'm just wondering.
Oh, yeah, I mean, that depends on system by system, but the level of agreement is pretty good. For example, let's go to this one. So they had this example of zebrafish like stripes. So they, I think, ablated some region and showed how they heal. And this is the simulation. So I mean, your initial condition can match your experimental condition. And you get some estimate of this diffusion coefficient and so on. Then you basically get similar behavior of these pigment cells. So
that's from the condition where there is already some kind of pattern and it's kind of recovery pattern.
yeah, so in this case it was recovery. But I mean, you can be, on simulation, you can be totally arbitrary, of course. And depending on, and also another thing is this boundary condition. So when you think about this animal, their topology is like you have these limbs and so on, like tubular manner. So if you put the right boundary condition, you can generate anywhere from like leopard spots to this coloring of panda. So it depends on just boundary condition and the parameters and so on. So actually, it's an interesting remark in this paper. They talk about this possible activator-inhibitor interaction in genes. We talked about this nodal and lefty and so on. So this is activator, that's inhibitor. And this is interesting. Note that the involvement of the reaction diffusion mechanism in some of the phenomena above has not been fully accepted by experimental researchers. So it's still under debate. Yeah.
You said the movement of the motor proteins was guided by diffusion. Are there any other things that can assist or just change the movement in any way?
Oh yeah, so there are two main pictures. One is called power stroke. Well, just the other is called Brownian ratchet. what I mentioned about this guided diffusion falls more in the category of Brownian ratchet. You have diffusion held by intermediate ratchets to direct the movement. How about power stroke? It's generated by some chemical event, and you create downhill free energy gradient. So if ATB binds and so on, you have conformational change. And if you make this downhill gradient, then you just move in a deterministic way, which is what you have seen in the case of this kinesin 1. So in this case, we cannot say this is like a ratchet-like mechanism. It's more like power stroke-like mechanism. But these two are not like mutually exclusive. I mean, at the like atomistic level, all this folding and so on is driven by thermal motion. In that sense, you can say this as really tiny progression of ratchet-like behavior. But in a more like bigger perspective, it looks like you are going downhill. But in the case of NCD, this guy, it doesn't have that active force generation. It just moves by in a diffusive manner. And that fits their function as well. If they generate too much force, then they will have hard time coordinating with other motors. They have to work as a group because they have to organize this mitotic spindle. Other questions?
The timing is good one because it looks like it's lunchtime.
Okay, great. Thank you.